Hello everyone, and welcome to today's live broadcast and keynote presentation. Genomics in the Clinic, a revolution for healthcare and medical research, presented by Dr. Marcel Dinger, Head of Genome Informatics at the Garvan Institute of Medical Research and Conjoint Associate Professors at UNSW, Australia. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. We're excited to bring you this educational virtual conference presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing the scientific collaboration and learning. Here's how this presentation will work today. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking at the support button at the top right or the Q&A button at the lower left. This is an educational webinar and thus offers continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your webpage and follow the process of obtaining your credits. Without further ado, I'd like to introduce our keynote presenter, Dr. Marcel Dinger. Welcome, sir.
Hi, everyone. It's a real pleasure to be able to present this this seminar today, and thank you, Susie, for the introduction. Okay, so today I'm going to talk about what I've learned over the last number of years in my role as both the CEO of Genome One and head of the Kinghorn Centre for Clinical Genomics on the impact of genomics both in precision healthcare and in medical research. So let me start at the beginning. Well, the beginning for you and me anyway. So that's the moment of conception, the moment when an egg cell from our mother and a sperm cell from our father unite. The egg contains 23 chromosomes from mum, and the sperm contains 23 chromosomes from dad. So together, and you may remember this from your biology textbooks when you studied, this makes our 46 chromosomes that make up our genome, the human genome. Each of those chromosomes is made from DNA. DNA, of course, is a polymer. So each chromosome contains hundreds of millions of these units, and these or letters that we call nucleotides, and they're represented as A's, T's, C's, or G's. So together, our genome contains more than 6 billion letters, and this is really what forms the core of what we're going to be talking about today. So I thought it was important to make that introduction. So this is all the information, effectively, that life needs to program a single-celled embryo that's created at the moment of conception to divide in a perfectly coordinated fashion into hundreds, thousands, and eventually billions of different cells as the fetus develops in the womb and into a human being. Every structure in our body, blood, cells, muscles, your nervous system, heart, kidney, bones, all of these things are all programmed and operated by information in our genomes. This program repeats with such remarkable precision, and as you can see in the case of the identical twins here in the bottom of this slide, that it runs with almost complete reproducibility. So it's not just even just eye and hair color, but even down to the finest details of where and when hair grows and how it ultimately recedes, and how our skin responds to the environment is all ultimately encoded in our genome. And in the context of disease, when our DNA is damaged at some point in our lives, that's how diseases such as cancer and the like can eventuate. Now, the information in our DNA is also what makes us different. Of those 6 billion letters of information, about 3 million differ between you and an unrelated individual. It's these differences that make us unique. It's also these differences that can cause disease, indeed virtually every disease, or our response to disease, is actually encoded in our genome. And so this is really quite remarkable, and of course you can also see that in the cases of severe disease, it can even just be one damaging mutation that can cause quite remarkably diverse sorts of diseases. So although our understanding of genetics and the principles of inherited characteristics date back, about 100 years, back in the days of Mendel, and which you'll be familiar with, we've understood the structure of DNA for just only 50 years. And it's only very recently that humankind has had the ability to actually sequence DNA and work out the order of what these letters are. At first, just tiny fractions or small parts of genes were able to be sequenced, but now it's possible in the 2000s, and really only in the last 10 years, to sequence the whole genome. At this stage, we only know a tiny fraction of what the genome actually means and what it does, but the fact that you can read a person's complete genetic blueprint or instruction manual, if you like, relatively quickly and cheaply is anticipated to have profound consequences on both the choices that we make in our lives and how we interact with the health system. So this is really the beginning of this new era that we refer to as genomic medicine, and potentially heralds a whole new era in healthcare more broadly. So putting this into context, in the last, this was a graph of disruptive technologies that came from the McKinsey Institute a year or two ago, and you'll see that many of these technologies you'll be familiar with, like the mobile internet and advanced robotics, the cloud, and self-driving cars, and those sorts of things. But you'll see there at number 7, next generation genomics as being one of the most influential technologies or most disruptive technologies that we can expect over in the coming decade. And actually, just advance the slide. So you'll see that by 2025, it's anticipated that next generation genomics will be in the order of $1 trillion industry. So because of of the list of disruptive technologies, the reason this one is so significant is it's actually the only one that has a real impact on healthcare, which makes it especially important. So this has been reflected in government investment around the world in genomics. In Australia, where I'm from, somewhere around in the order of around 100 million has been devoted towards developing genomics in healthcare. But around the world, the figures are actually much greater, with in the order of hundreds of millions spent in the UK, billions in the US and China, and somewhere around close to 700 million euros in France, all towards an investment in understanding the role of genomics in healthcare. So what are the opportunities? What is it that makes genomics such so special in its context in healthcare? Well, first of all, when we look at I guess its most immediate impact is clearly in the diagnosis of rare and inherited disease. Perhaps makes sense, if you think about genetic disease and we're able to sequence a genome, it follows that we're going to become very, very good at diagnosing genetic disease if we can, in a single test, read this information. So we go to cancer, and the opportunities there for sequencing tumors and being able to, in fact, cancer is also ultimately a genetic disease. It's a case where the cells in your body are starting to divide in ways that are uncontrolled because the genome is basically being altered in a way that it's not supposed to. And by being able to define the exact biochemical basis or molecular basis for what's happening in that tumor, we're able to much better target its treatment. At least that's the direction that the science is going in. And then finally, there is a view that that the capacity to be able to sequence a genome in advance of having disease will have the most profound consequence of all in healthcare, where it'll be able to anticipate the susceptibilities or predispositions that individuals might have for cardiac disease or cancer or adverse reactions to drugs. So, unlike other molecular tests, what we see with genomics is, in fact, that it has an impact across your entire life. So, right from from planning to have a child in the context of carrier screening and working out which individuals may have a recessive disease that if that comes together would be expressed in a child, can be anticipated, all the way through to being able to read and understand the information that happen at the point of in the fetus, and following that, the child is born, being able to predict what potential health problems might emerge early on in life that can be potentially prevented. So the area that we're most closely in is in the area of pediatric disease, and at this time but we certainly expect that in the future cancer diagnosis, and even eventually in the context of a molecular autopsy, understanding why it is that somebody died. So what's made this all possible is really this dramatic shift in technology that's made it possible to sequence a genome for far more inexpensively than it was even just 10 years ago. So 10 years ago, it cost somewhere in the order of $100 million to sequence a genome, whereas following this development of new technologies and the introduction of massively parallel sequencing in around 2007, we start to see this hyper-exponential decrease in the cost of sequencing the whole genome. And then in 2014, with the release of the Illumina instrument, the HiSeq X10, it became possible to sequence the genome for a base cost of around about $1,000. So this brought it into the same context as other sophisticated medical tests such as MRIs. So it's very unusual to see a technology where you get a 2,000-fold reduction in cost in the space of just a decade. So this is the instrument that made it possible, the Illumina HiSeq X10, which was released in 2014, as I mentioned, which can sequence genomes not only at a relatively low cost, but also at large scale, about 18,000 a year or about 300 per week. So it transforms, this technology really transforms the entire genetic testing market, because it makes it possible that all of these previous types of individual genetic tests, such as cytogenetics, MLPA, individual gene sequencing, become replaced effectively just by a single test. So rather than, this is transformative, because for a geneticist who's trying to anticipate which test to use, you no longer really need to do that. You just do one test and you'll have all of the information, and then it's really just a question of what part of the genome you need to look at. So this is transformative in the clinic. So our role in this, and just to set the scene, was that the Garvan Institute was one of the first 3 sites in the world to take on this technology together with the Broad Institute and Imagery. So our centre, the one that I work in, is the Kinghorn Centre for Clinical Genomics, and it was established in about 2012 and made use of both through philanthropic and state government funding. And the mission of the centre, which I think is one of only few of its kind in the world, is really to bring genomic information into whole-of-life healthcare. So it has a very strong translational focus. So the objectives were really to translate the technology of whole genome sequencing from being a research tool and into the clinic and ultimately with a view that this will become a routine diagnostic test. And of course, the other aspect that I'm going to touch on in this talk is also how we can bring together clinical information and be able to utilize this in a research setting as well. And I think this is a significant paradigm shift where we've traditionally seen a real separation between data that's generated in clinical context and information that's generated in research context. I think what genomics is going to do is going to bring these fields much closer together. So our centre, as I said, started in 2012, and really the only thing I want to point out here is just the distribution of individuals in this translational process, where we started out, of course, with bioinformaticians and some technicians to run the sequencing instruments. But as we grew, it becomes, as you can see, far more heavy towards utilization of bioinformatics and software engineering, and eventually also deep clinical integration to be able to take a technology that was really developed in a research environment and being able to translate it into a clinical environment. And what we've achieved now, I think, through this is an ecosystem where clinicians and researchers can work very closely together. So if we take a more traditional look at how the separation between medical research and industry and pathology and clinical care formerly were, where we used to, in the context of, for example, identifying a new genetic marker, you would have a patient cohort, you would research this cohort and sequence it, and then perhaps identify something new. This would get published, and then at some point, industry or pathology might take this on and show that this new marker has clinical utility, and then that might from there emerge into a genetic test, and then from there that could become used and translated into the clinic. And I think what we're seeing now, and in the enabling of precision medicine, this particular model is not one that is really going to, that can't really endure for the realization of precision medicine. And the reason for this is that there are now so many different things that you can test for that it's very unlikely, or it becomes so infinitely costly to be able to run through this trajectory across thousands or tens of thousands of different genes. And in fact, what I think we'll see is these fields coming much more closely together. And I think really I guess the philosophy of our centre is to take away these boundaries that formerly existed between these industries and start to bring these things together into a common ecosystem. So we're rather of thinking of a patient research cohort, the research cohort also becomes in parallel the clinical trial cohort. The basic research becomes translational research. And when you identify a genetic marker, it becomes the genetic test. And in an environment where this becomes possible, you get this remarkable efficiency in being able to translate research information into clinical outcomes in a far more efficient way than was ever possible before. And at least in part, this is what we've been striving to achieve. And you end up with this very virtuous sort of a cycle where research can be translated almost immediately and transferred into clinical care in the same setting. So what makes this possible, of course, is that you need to operate in a very different environment, first of all, closely with clinicians, but also secondly, that the test data that you're creating is done in an accredited laboratory so that information can actually be returned back to the clinic. So this is what we see as a future model for how healthcare may look in a few years into the future. So the idea that you're taking, if you look at the top half of this chart, and you can see that there is information from demographical information, clinical information, EMR data, and so on, all kind of coming together into a common database, if you will, or into a common repository that can be shared. And the key things to make this happen, of course, is that consent needs to be done appropriately from the outset. So regardless of whether an individual is a research participant or a patient, that they are consented for information to be used in this way. So you also need, of course, the clinical data to be entered into a way that it can be shared and manipulated and in a format effectively where you can use it for analytics. And what we then expect, and if you look into the middle part of this, is that it's really the genome that forms the backbone of the information that all of this loads onto, and that from there we can use new machine learning algorithms to optimize clinical decision making in real time. So what we're really saying here is that in the future, what we expect to see in the realization of precision healthcare is effectively where all of the information that's available about all patients in the world is actually able to be used to inform new decision making. So rather than a research study necessarily informing the next level of evidence-based clinical care, is that evidence base is actually being built about in real time. So that, for example, the responsiveness to a particular drug is immediately known from its use around the entire world. And by matching this back up with genetic information, when a new patient arrives, that you're able to go, okay, this drug is going to be really effective on this individual, or it's not going to be effective. And that data is actually available to clinicians in real time. So we're not there yet, but certainly what the ambition is of our centre, and and this was really the first steps in this journey, was to be able to, I guess at its most basic, for a clinician to be able to request a whole genome sequence for an individual and to be able to get a pathology report in return that they can then act upon to guide the care of that patient. It's a very complicated process that happens through this. I won't go into this into great detail, but effectively there's a counselling step, and then of course there's the acquisition of clinical information and DNA, which is then sequenced, analysed through a sophisticated bioinformatics pipeline, and then ultimately interpreted and placed into a report. So embarking on this 3 or 4 years ago was a significant challenge, and there were a lot of things that needed to be sorted out. Not the least of which was to have a mechanism through which blood samples can actually be taken from anywhere in the population and returned into our laboratory. We did this by partnership with an existing pathology provider. We of course established the high throughput genome sequencing with the X10, which we took to scale, and were able to start generating data at quantities of around about 1,000 genomes per month. We needed a mechanism to be able to create, to capture phenotype. So there was a platform, which I'll describe in a bit more detail shortly, that enabled a standardized mechanism for capturing phenotype. And of course, quality was fundamental in this. We had to be able to demonstrate that this was fit for purpose for clinical use, and of course needed to have this backed up through clinical expertise that we partnered with the public pathology provider. Our diagnostic rates proved to be quite exceptional with whole genome sequencing, and you consider that the former technologies were able to diagnose perhaps 10 to 12% of individuals. What we found with whole genome sequencing is that we're able to diagnose around about 50% of cases, and these are often the most difficult of cases of individuals that are affected by rare genetic diseases. Quality management becomes fundamental. These are very different processes than what you're familiar with in a research environment, being able to apply this into a clinical setting as well, and leading ultimately to accreditation and ultimately also commercial delivery through a spin-off company that we formed called Genome One. So effectively, what we achieved through what is a very harmonious relationship that's been developed between the Garvan Institute and Genome One is to enable this cycle that I described before, and it really puts Genome One in the position where it's able to acquire clinical grade genomic information and use it for either diagnostic or research purposes. So for the remainder of my talk, I'll just talk a little bit about how we approach this in the context of rare disease diagnosis, but not forgetting that genomics has potential across a very wide range in healthcare. So why rare and inherited disease? So in Australia, at least, it's about 1 to 2% of Australians are born with a severe genetic disease, for many of which are undiagnosed. Diagnosis can guide treatment. It's a very valuable process for patients and for families. And as I said, the diagnostic yield can be very high in this space. And of course, because this is a relatively a small community, still small but very significant community, it's possible to implement this in the health system by having this focus in the clinical genetics arena. So, and of course, there is a very strong economic case for the introduction of the technology. So we were able to make some real personal impact on individuals, where, as you can see here, this is the mother of one of our families that we were able to do a diagnosis for, that had gone through really a remarkable journey of negative tests, effectively, and treatments that were ineffective, to eventually come across what we were able to achieve with whole genome sequencing and be able to provide a diagnosis, and it had a huge impact on this family and has had on many others. So coming back to what makes this so difficult and where the challenge sits today in terms of being able to generate the data, is something that can be done now quite routinely with an instrument, but the interpretation of these 6 billion nucleotides of data is still a huge challenge. So between each of us, there's about roughly 3.2 million variations between us. But each of us actually carry, around about 100 or so genes that could be considered as being pathogenic, which is remarkable and perhaps somewhat unexpected. But it makes it actually very difficult to sometimes identify what the cause of a disease is, when in fact a healthy individual also has a number of defects in genes that don't seem to actually have such a direct effect. So this filtering that we go through is really what the magic is that happens at the diagnostic level and in the interpretation laboratory. So we take these 3 million variants and we run them through a number of different databases and filters and computational algorithms, and ultimately end up with this number between 50 to 100 or so of different mutations that we then try to analyze and try to connect with the disease. One of the really important steps in doing this is the use of phenotype. So, as you can see in this photo here, phenotype is a core driver for how we actually interpret variants. So, what that means is that we need to have quite detailed information about the characteristics of the disease we're looking for to be able to get the real short list of variants that we actually believe are causative in the disease. So in this particular example, and these are two unrelated boys, you can see that they have some very similar characteristics. So they've got the downturned corners of their mouth and low set ears and a couple of other qualities there that look similar. These two boys are unrelated, but some people see this photo and they think they could almost be brothers. So, but in fact, what happened was that this was a case where one of the boys had a query as a potential gene that was causing this so-called PAX1 gene that was causative for this disease, but it had never been seen before. So, without having another case, it made it very difficult to be sure that this was the correct gene that was causing the disease. And what happened in an ad hoc fashion is that this was presented at a conference, as the story goes, and there was somebody else in the audience who said, oh, I've got a child and a boy in my clinic who looks like they have similar characteristics. And sure enough, they tested that same gene and they had the identical mutation. And so a diagnosis was made. Now, in a case like this happened effectively by serendipity. And what is now trying to be coordinated is how we can actually bring these types of cases together so that we can more effectively and more efficiently diagnose rare diseases. So at the moment, the world of health, this kind of looks like this, where geneticists will often speak to each other and may just phone one another up or meet at conferences, and which of course is a very, as I said, a fairly random and ad hoc way for a diagnosis to be achieved. So what the aim today is to be able to try and share phenotypical information in an automated way, in the same way that we can share genetic information. So unlike the genome, which has got, of course, a very standard language, A's, T's, C's, and G's, are very easy to represent, and it's a common way to represent data. For phenotypes, that's not the case, and in fact, phenotypes can use descriptive terminology that's often non-standardized. So what the real aim is to be able to really understand the genome, we need to be able to extract this information from what we call the phenome, which is the collection of all attributes that we have in all of our physical and biochemical characteristics that are expressed by our genome as our phenotype. And ultimately, we want to be able to bring this information in terms of how the environment, in fact, impacts on phenotype as well. So it's the bringing together of this information, but to do this, you need to be able to accurately record phenotype. So to this end, we've developed a platform, which we refer to as Patient Archive, which effectively uses a standardized terminology set from the human phenotype ontology that extracts, that can then be used to standardize clinical notes and then be able to share this data. So I'll just talk to this briefly. The key features of this platform and technology is that it's able to take clinical summaries and notes and be able to extract the standardized human phenotype ontologies from them. So, for example, terms like say, large head would get turned into macrocephaly, for instance, and so big head would also be called macrocephaly. And so you can sort of start to take normal language and it can be translated into this clinical terminology. And then this is what the information that can then be shared into databases, and then other clinicians can then use a common language to be able to interact and potentially match patients together. And that's really the idea of this technology, and it's been working very effectively in this context. So the view that we're trying to build towards is actually trying to integrate genomic and phenomic information, either in a centralized or in in some sort of a federated manner, where this is actually where you start to build networks of genomic and phenomic information. And from that, collectively, we can both better understand what characteristics can be learned from the genome, and reciprocally become more effective at diagnosing disease. So this is I guess the view that we can develop through this is that an individual has their phenotype captured, the genome sequence, and this information is centralized. The information that can be interpreted clinically can then be used for clinical action, and the information that we don't yet understand can be used to research and to identify new knowledge, and therefore building new gen-phen or genotype-phenotype associations with the genome. So the idea is that progressively we start to learn more and more about how the genome informs the phenome. And of course, this creates a huge big data problem. So just to finish my talk, I'll just talk a little bit about where I see this going in the future. So certainly genomic pathology is really just the beginning of this journey, and in the context of rare disease, of course, is where we started, but there are many other major diseases that have a massive impact on the health system, including, for example, kidney disease and cardiovascular disease, that have a strong genetic basis for them, and where knowledge of genetic information can potentially inform patient management. But really, as I said, it has this impact across all of life, this whole of life care impact, where at some point in the future, perhaps, when a genome is sequenced, perhaps at birth, where that information is then requeried again and again over the course of a person's life to inform different parts of what's happening at that stage. So, for example, if you get cancer, then the information might be used to anticipate what the best treatments might be, or in the case of an adverse drug reaction, or before having a drug, that you're able to predict which drugs are going to be effective or which ones you may have an adverse reaction to. So I guess the interesting thing that this creates about the genome is that we end up with this graph where the cost of the genome continues to come down and the value of the genome continues to rise. So as we sequence more genomes and more genomes are connected to health information, we start to learn more and more about what the genome can predict, and we become, I think, more powerful at being able to extract information from the genome. So at some point we'll reach this cross where these lines intersect, where you end up with a far where, of course, you know, this line has already been crossed in the context of rare disease, but it will start to cross at as time goes on into more and more different types of diseases, and eventually the anticipation is that there will be value in any individual to have their genome sequenced. So the figure there on the right just shows the number of genetic loci that have been connected to a disease. There's a complex disease like schizophrenia, and you can see just even over the last 5 years, where in 2009 almost nothing was known about the genetics of schizophrenia, and that by 2014 hundreds of genetic loci have been connected to this disorder. So, and the same story holds across many different diseases, and we're learning more and more about these all the time. So we expect, therefore, that genomics will expand into what we call whole-of-life healthcare management, where we're really a part of healthy aging and being able to optimize not only what drugs you take, but what food you eat and what other choices you might make in your lifestyle to be influenced and impacted upon by genomic information. And we are seeing this rapid growth in our understanding between genetic makeup and characteristics. And I think as the data continues to grow, we'll be able to become better and better at predicting this. And you know, there are data already been shown that the capability to be able to even predict detailed features of an individual's face purely from and other characteristics like height and weight, potentially from genomic information, which is quite remarkable when you think about what this holds in the future. So but it's a long journey ahead before we're there, and this is a nice figure, I think, that really illustrates exactly what's happening with this. So let's see if I can use an arrow here. So in the top left corner of this diagram, you'll see that monogenic disease, which is where we where there's the most readily accessible mechanism to be able to diagnose disease, sits in this area. This is where a single mutation causes an extreme effect. So that's what we see in Mendelian disorders. But as we go down the chart from top to bottom, you end up going towards more complex diseases where there is a greater and greater environmental component. And so we accordingly get diseases or genes rather that can have mutations in them that will have a different impact on different individuals. And that can depend on what other genes they have and the mutations that they carry, and it can also depend upon what environments they were exposed to. So a good example of this is like the BRCA gene, the BRCA gene, which causes a high predisposition to breast cancer. But not everybody that carries the BRCA gene gets breast cancer. In fact, only, maybe it varies from even different ethnic backgrounds from 50 to 85 or so %. The reason for this is thought to be, some of it may be environmental, but certainly some of it will be because some individuals carry other protective genetic variants that prevent the cancer from forming. So this is really what is now trying to be understood, and I think will be understood as we learn more and more about the genome. So to achieve this, and this is really the advent of a whole new field, is the application of machine learning and deep data analytics and genomics. And indeed, there are already companies that have formed that are starting to take this on scale. These are seriously big data problems, and will no doubt require new algorithms to be developed, and I expect new supercomputers to be built around the world to be able to manage and predict this information, because the scale of the data becomes really quite remarkable at this stage. And at this point either, we don't fully understand how information is encoded in all parts of the genome. In fact, our understanding of the genome is really restricted to perhaps 1 or 2% of the genome that we are able to interpret accurately. And much of the other parts of the genome, the so-called non-coding DNA parts of the genome, much of this remains very challenging to interpret. So the national initiatives that are moving forwards, for example, in the context of things like Genomics England, we're sequencing 100,000 genomes. What we're starting to see now is this development of mechanisms to both to scalably store and share data. But in parallel to all of the genomics technologies that we're seeing, we've seen emerging, something else that's happening in parallel is this explosion in mobile health monitoring technologies, where it's now become possible to measure a lot of actual, and some of these technologies are actually clinical grade, which you no longer need to necessarily go to a clinic to be able to take every type of measurement. And in fact, this opens up an entirely new opportunity where instead of just getting a snapshot of your health information, you can actually get certain types of health information monitored in real time. So this goes well beyond just pulse rates, like you might do with a Fitbit or something, but what you can actually do with real-time ECGs and systems for monitoring blood counts and neural activity and so on is becoming more and more vast. And this longitudinal information, once correlated again with the genome, may become incredibly powerful for learning and anticipating disease in the future. So we certainly see that genomics is poised to catalyze a revolution in analytics and decision support and potentially even mobile health. And so this is, I think we're really at the start of a remarkable journey in a transformation in healthcare that I think will be at its base driven by genomics, but deeply connected into many other new technologies that are emerging. And we see, certainly from our perspective, that this is going to impact across the entire health system. So this isn't just for driving diagnoses and research, but could actually have real public health impacts, as well as allowing the health economics to actually drive decision and policy making in ways that were never possible in the past, and it may just provide at least somewhat of an answer to the continuing and burgeoning growth of health budgets around particularly first world countries around the world that are becoming rapidly unsustainable. So to finish with, these are really the conclusions that we see from this, is that whole genome sequencing is a single test that can potentially diagnose any heritable disease, it can tailor drug choice, and it can predict health outcomes. We do absolutely anticipate that a test of this power, particularly as its information value continues to increase, will ultimately become part of our health record and will become a central tool in decision making in the clinic. There's still an enormous challenge in taking this information apart, and I think, you know, what we call decoding the genome is a long way to go. There are millions of variants that we do not yet understand what role they have on our health and really what they do. So that's something I think that will emerge through, as I said, advanced information analytics. So, but the other, I think, interesting observation around all of this is the development of this, and what we've really seen in action is this or what I call this closing of the gap between the clinical and medical research communities in a way that's never has never really occurred before, and where we're now using the genome as a common backbone, or as a substrate from which both healthcare and research seem to be mutually benefiting and growing from. And I think this can really lead to is actually a fundamental component of what will enable precision medicine in the future. And with that, I'll finish, and I'm happy to take any questions. Thank you.
Thank you so much for that informative presentation, Dr. Dinger. We want to begin our Q&A segment. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we are unable to get to your questions due to time constraints, Dr. Dinger will be answering submitted questions via email. So let's take our first question. An audience member would like to know, you described the use of genomics mostly for diagnosing diseases. What can the genome tell a well person about their health?
Question. So I think that that's a great question, thanks. So I think at this stage it's still early days in terms of the information that's valuable to inform the health of most healthy individuals. However, clearly we can already report valuable information in the context of cancer predisposition, cardiovascular disease, and in another domain, pharmacogenomics, which is predicting both the metabolism and reaction to drugs and pharmaceuticals. So I think that's probably where it's starting today, but we can certainly see that in the future. Areas like carrier testing, I think, are very valuable, and being able to anticipate and predict other diseases. And many of these have actually got really valuable clinical interventions. So, you know, obviously in the case of things like cardiac disease or cancer, where more regular screening, for example, might be recommended to somebody who has an unusual predisposition to a disease. So we're certainly starting to see this emerging.
Thank you so much, Dr. Dinger. We have another question from one of our audience members. She'd like to know, as I understand it, only a small amount of the genome can be used clinically. Why did you choose the whole genome instead of exomes or panels?
Yeah, interesting question. So it certainly is the case that using exomes or panels are very effective today in diagnosing some types of disease. Our motivation really for using whole genome sequencing was that it provided an opportunity for taking all of those negative cases, the ones that weren't able to find a diagnosis, and then being able to put that information back into the research environment. I guess the rationale is that if we don't start out by sequencing the entire genome, we are ultimately limiting ourselves in what we're ever able to learn. So to build this ecosystem that I described between healthcare and research, I think we need to simultaneously gather information that has both clinical and research value, and the whole genome is a very practical way of doing that. And of course, the price gap between the exome and the genome continues to become smaller and smaller, and so there is, of course, an anticipation perhaps that we will eventually see that whole genome sequencing will be a very similar price to and cost to exomes.
Thank you so much, Dr. Dinger. As we end this presentation, do you have any closing remarks you'd like to share with the audience? This has been a tremendously informative presentation.
Sure. Look, I'd just like to thank everybody again for taking the time to listen to this presentation, and I hope that you found this an interesting way to start thinking about how healthcare and medical research might really start to have a deeper intersection, and how this future of precision healthcare is really requires the efforts from both of these industries to come together to really realize. Exciting times.
It is pretty exciting times, and we want to thank you again, Dr. Dinger. Thank you, audience. I'd also like to thank LabRoots for making today's educational webcast possible. Today's webcast will be available on demand viewing through August 2017. Keep an eye out for an email from LabRoots alerting you when this webcast will be available for replay. We really encourage you to forward the announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope to see you next time. Goodbye.